Okay guys, today we're going to talk about nerve cells, neurons and synaptic transmission and how that can result in an action potential being formed in the postsynaptic neuron and therefore a nerve impulse. So if I just draw a typical motor neuron, so these are the dendrites, this is the axon, and then at the end you'll have the synaptic bulbs at the end of the axon here. So the axon is myelinated, so the cells that make up the myelin sheath are called Schwann cells. This actually happens during fetal development, where the Schwann cells wrap around the axon and they make a hydrophobic material called myelin, hence it's called the myelin sheath. So Schwann cells wrap around the axon many times with their cell membrane. So there's lots of phospholipid being wrapped around the axon. So this is called an electrical insulator. So we could say electrical insulator, and that's going to speed up the rate at which the nerve impulses passes down the axon of the motor neuron. Now the gaps between the Schwann cells where there's actually no myelin sheath are called nodes of Ranvier. So here, these are nodes of Ranvier. Now that's going to allow the nerve impulse to jump from node to node, and that's called saltatory conduction, as the nerve impulse passes from the cell body, which is where you would find the nucleus, down to the synaptic bulbs, which is at the end of the axon. So these are the synaptic bulbs. So this would be the nerve impulse. And because it jumps from node to node, we're going to call that saltatory conduction. Now, the cell body, the cell membrane of these dendrites, these are called the postsynaptic membrane. So you're going to have synaptic bulbs from a presynaptic neuron. So imagine we had this scenario where we had the synaptic bulbs from another presynaptic neuron. Okay, so the dendrites would be the postsynaptic membrane. And the synaptic bulbs of the previous neuron would actually be the pre-synaptic membrane. So the gap between the presynaptic membrane and the postsynaptic membrane on the dendrites would be the synapse. So the gap obviously would be a lot smaller than I've drawn here, but I'll just put this here just to show you that this would be the synapse between maybe this part here, the presynaptic membrane and the postsynaptic membrane. So this is the gap through which neurotransmitter was diffused by, from higher to lower concentration across the physical space once they'd been released by exocytosis. So what we're going to do is we're going to take this part of the diagram up here, and we're just going to draw that in a bit more detail. So this is going to be the synaptic transmission. So if I just do the synaptic bulb, so this would be the presynaptic membrane. So I'll just put presynaptic membrane, which is that membrane there. This is the presynaptic neuron, and the gap here would be the synaptic cleft. And then what we're going to have is the cell membrane of the dendrites, which we now call the postsynaptic membrane. So we'll do that at the bottom. So this is postsynaptic membrane. Now, as the nerve impulse gets to the end of the synaptic bulb of the presynaptic neuron, it's going to be a series of action potentials that go down the presynaptic membrane. And what that's going to do is it's actually going to open calcium channel proteins. So these are voltage gated, and these are going to open at plus 40 millivolts. So as the action potential, which is plus 40 millivolts, gets nearer to the presynaptic membrane, it's going to open these calcium channel proteins, which are voltage gated. And when these open, it's going to allow the entry by facilitated diffusion of calcium into the cytoplasm of the synaptic bulb. Now, the increase in concentration of calcium, what that does, it causes secretory vesicles that contain neurotransmitter to migrate with and fuse with the presynaptic membrane. So as these fuse with the presynaptic membrane, the neurotransmitter is released into the synaptic cleft. Now, you've got to be careful here because it is diffusion. So we're going to get higher to lower concentration and diffusion down that concentration gradient across the synaptic cleft. And a typical neurotransmitter would be acetylcholine. So I'll just put acetylcholine, and that would be a cholinergic synapse. Now, on the postsynaptic membrane, there are receptor proteins that have a complementary binding site. The shape is complementary to the shape of acetylcholine. So these receptor proteins on the postsynaptic membrane are going to bind the neurotransmitter, like acetylcholine. And what that does, it opens sodium channels in the postsynaptic membrane. So this is going to allow sodium to move by facilitated diffusion into the cytoplasm of the dendrite. So these are ligand gated because it relies on the receptor protein on the postsynaptic membrane binding the neurotransmitter, which then opens the sodium channel. So these ones here wouldn't be voltage gated, that we'll have a look at in a minute on the axon membrane. These are what we call ligand gated. Now, the membrane potential at this part of the neuron is going to be at resting potential. That's called minus 70 millivolts. So this is called the axon hillock. And this is where we're going to get possibly the first action potential. So the resting potential is is minus 70 millivolts. Now, if enough sodium comes into the cytoplasm of the cell body, because there's enough neurotransmitter binding to the receptor proteins, it might cause that minus 70 millivolts to get to minus 55 millivolts. So it starts at minus 70 millivolts resting potential. There's a stimulus, which is binding of the neurotransmitter to its receptor. That might open enough sodium channels, so we get sodium moving in by facilitated diffusion, and it might cause the minus 70 to raise enough to get to the threshold of around about minus 55. Now, if we do get to the minus 55 millivolt threshold level, or whatever it gives you in the question, it'll be around about minus 55. Sometimes it's a bit less or a bit higher. It's going to cause at this part in the membrane. So I'm going to zoom in on this part here. So this is the axon membrane. And remember, it started out at minus 70 millivolts. That's resting potential. But all of a sudden, this has got to minus 55. Well, in that axon membrane are voltage-gated sodium channels. Now, they're voltage-gated because all these open at that position at the threshold value. So I'm going to say that's minus 55 millivolts. So we've got sodium moving in to the cell body across the postsynaptic membrane. The resting potential of minus 70 millivolts has now been raised to a threshold value. The sodium channels at this point on the axon hillock in the axon membrane are voltage gated, so they're going to open. Now, one of the proteins that is in this part of the membrane as well is called the sodium potassium pump. Now, what that does, it pumps out 3 sodium ions for every 2 potassium ions that come into the cytoplasm of the axon. And it does require ATP, so it's very active. But because it pumps out the sodium ions, there's going to be a higher concentration of sodium in the tissue fluid outside the axon. So we've built a concentration gradient higher on the outside of the axon, lower in the cytoplasm. Now, because there's a concentration gradient and the sodium channels are voltage gated and they open at threshold, it means the sodium can now diffuse into the cytoplasm of the axon. So this would be the cytoplasm in this part of the picture. And that's going to reverse this membrane potential from the threshold of minus 55 millivolts all the way up to plus 40 millivolts. So the membrane potential is always the charge of the cytoplasm in relation to the outside charge in the tissue fluid. So if there's loads of sodium ions that diffuse in now by facilitated diffusion, it means actually the charge in the cytoplasm is plus or positive in comparison to the outside of the axon. Now the sodium channels actually close at plus 40 millivolts, but we've had depolarization of the axon membrane because it's gone from minus 70 millivolts, which is polarized, to being depolarized, so it's the opposite, and it's gone from minus 70 up to plus 40 millivolts. Now at plus 40 millivolts, what we actually now find is, so I'll just do the axon membrane again here at the top. There's other channel proteins, and these are now potassium channel proteins at that point in the membrane. And they open at plus 40 millivolts when the sodium channels close. So because the potassium, remember the sodium potassium pump was pumped in, 2 of them for every 3 sodiums that get pumped out, there's a concentration gradient now of potassium ions, so there's more of them inside the cytoplasm of the axon versus the outside. And because these open at plus 40 millivolts, the potassium channels, the potassium is then going to diffuse out of the cell, again by facilitated diffusion. So that's going to reverse the membrane potential from plus 40 millivolts, and it's going to fall back down as the cytoplasm loses these positive K plus ions. Now, because these do not close at minus 70 millivolts, they actually stay open for a bit longer. It allows more potassium to move by facilitated diffusion out of the axon cytoplasm. So we actually end up with not just a return back to minus 70 millivolts, which is where we've repolarized the membrane, but because they stay open for a bit longer, we actually get to minus 90 millivolts, which is called hyperpolarized. Now, the fact that the membrane at this point becomes hyperpolarized is actually really important because it's going to allow saltatory conduction, the jumping of the nerve impulse from neuron VA to neuron VA to happen in a single direction. And we're going to see why next. So if I do another axon membrane here, and I put in our myelin sheath with our nodes of neuron VA. So this is the myelin sheath. These are the nodes of neuron VA. Okay, so there's no electrical insulator at the nodes of neuron VA because there's no myelin, there's no Schwann cells that are wrapped around the axon at this point. So this is the axon cytoplasm. Okay. Now, imagine we had at this point on the axon membrane plus 40 millivolts. So remember, it started at minus 70, it became threshold minus 55, and then it changed to plus 40 millivolts due to depolarization. So this would be an action potential. And we know we've got some sodium in the cytoplasm here because that came in during depolarization. Now, the sodium doesn't just stay there in the cytoplasm of the axon. What's going to happen, it's going to diffuse down the inside of the axon from higher to lower concentration. Now, the next node of Ranvier is minus 70 millivolts. That's resting potential. But the diffusion of the sodium down the cytoplasm of the axon is going to cause that to increase up to maybe minus 55 millivolts. And we know that's the threshold potential. Therefore, at this point, the sodium channels are now going to open, which is going to allow the sodium to flood in via facilitated diffusion, and that's going to create the next action potential at the next node of Ranvier. Now, likewise, the sodium, as it floods in through facilitated diffusion, through depolarization, which causes the action potential, that is now going to diffuse down the cytoplasm. Remember, that was minus 70 millivolts. That gets to minus 55 threshold. We're going to get depolarization again through the opening of the voltage-gated sodium channels. So that's going to create the next increase to 40 millivolts. So that will be the next action potential. Okay. But remember, we said that after the action potential, you get repolarization and then you get hyperpolarization. So that membrane here further back is at minus 90 millivolts. So when you get the second action potential and the sodium floods in by depolarization, some of it might actually move this way, but because that membrane is minus 90 millivolts, it's hyperpolarized, you can't reach another threshold potential. So this would be in hyperpolarized state, whereas this node of Ranvier might be minus 70 millivolts, which is a bit higher than minus 90. So minus 70 is a lot easier to get to threshold than minus 90 would be. So when the sodium moves in here at this point through depolarization, okay, it diffuses down the axon, and it's easier to get to threshold at the next node of Ranvier further down. So it's going to cause depolarization at the next node of Ranvier, but not at the one further back, because that's hyperpolarized, and that's in what we call the refractory period, where you can't get another action potential. So what's going to happen is the nerve impulse is going to jump this way, okay, from node to node in a single direction along the length of the axon of the nerve cell. And again, we call that saltatory conduction. Now, when you get a hyperpolarized membrane, so imagine this example further back here, we are going to re-establish the resting potential because the sodium-potassium pump, remember, continues to pump out the 3 sodium ions and pump in the 2 potassium ions to re-establish the resting potential. So when you get one action potential here, you will always get another one at the next node of Ranvier, and always another one at the next node of Ranvier, which is actually the nerve impulse as it passes down. Now, one good way of remembering, if you just go a bit further back up, that 3 sodium ions are pumped out by the sodium-potassium pump and 2 potassiums are pumped in. If you think of a sodium ion, it's got 3 parts to its symbol, so N, A, and plus. So 3 sodium ions are pumped O-U-T out. And potassium has got 2 parts to its symbol, a K and a plus. So 2 potassium ions are pumped 1, 2 in. So K plus, 2 potassium ions, IN in, Na plus, 3 sodium ions pumped OUT out, which matches what we had at the top.